આપેલું ફરી જાય પછી જાવ કોઈ છે કે લાઈવ Hello everyone. Am I audible? Am I visible? Please write in the comment section. Sorry guys, streaming is little bit lagging. Okay. Okay. So yes, I think now it is okay, and welcome to the another session, which is a Saturday night with Dr. Nikunj with me on the behalf of VASISHTHANEET dedicated NEET Academy. Guys, in the previous session, in the previous class, we were discuss about the various facts and various topics such as the nucleic acid, the composition of DNA, and so on. So in this particular session, today's topic, a topic of interest is what? A topic of interest is the enzyme. We will first discuss and look on the enzyme. After finishing the enzyme, we will go and discuss about the another topic, yes, which is a cytoskeleton. And after finishing the cytoskeleton, we will move on the last topic of the day, which is the Kern-Class theory. Here in this case, maybe these two topics are little bit newer for you, but these are the topic which having a very important stance in the examination. So let's get started. So the first topic of our interest is the enzyme. So let us move on the enzyme. Let's look the enzyme. What is enzyme actually? So enzymes are what? Enzymes are the component which is the biological catalyst, right? It is what? It is the biological catalyst. What do you mean by biological catalyst? See, in our body, there are lots of reaction, such as the photosynthetic in plant body, such as the cytoplasmic respiration in a human body, which is also known as a dark respiration. So all this reaction needs something which boosts the process, So that particular thing in our body, which is the catalyst for us, is the enzyme, right? So enzymes are what? Enzymes are the biological catalyst. Now the term catalyst means something which is acceleratable, right? The something who accelerate the reaction, something who accelerate the process or a speed of process, that is known as the catalyst. Here, this particular thing, which is the enzyme, used under various biological procedure. Okay, it is used under various biological procedure, and that's why it is known as the biological catalyst, right? So I think you people might get the idea. I'm so sorry guys. Yes, I greet Vinay. Thank you so much. Viraj, Atif, Ishwar, Patel, Akanksha, right? Patel, Twisha, thank you very much for attending this session. And the session is little bit actually the live streaming, and my voice or my actions are little bit lagging around 10 to 15 seconds, so I cannot answer you as immediate as you expect. So please, sorry about that. We will find a solution for that in the next session, but please focus in this session. So the enzymes are what? Enzymes are molecules. Normally they are chemically proteins. Okay, so what is the nature of enzyme? Enzymes are generally the chemicals which is protein. And what is protein? Protein is the macromolecule developed, formed by an monomer, which is the amino acid. And maybe you know that the amino acids are what? Amino acids are basically a component which compose the amino group at one place, and on the other hand we have the acid group. One hydrogen is there and one random group is there. So if you look carefully under this component, you will see that it is nothing but a derivative of methane, right? It is nothing but a derivative of methane. And also you learn that the proteins, basically the proteins are what? It is a heteropolymer. Okay, proteins are a heteropolymer. That means if a one particular amino acid present at a chain, the another or subsequent the amino acid will be different. It might be maybe the first one is valine, the another should be glutamine, another should be threonine, or anything else. We know about 20 types of amino acid. Actually we know about more than that, about 200 amino acid. It is very advanced actually, but the 20 amino acids are the workful, it is the functional enzymes, or you can say it is a functional amino acid present in our body, right? And they are responsible to construct, to formulate our enzyme. So enzymes are nothing, enzymes are catalyst, which is the biological catalyst. Biological catalyst means it is only work during the biological reaction and it will make it fast, right? So it is just little bit like accelerator as in the chemical reaction. You learn about the chemical reaction so well in your chemistry lecture. So you learn that the chemical reaction, basically what are chemical reaction? Chemical reactions are those reactions in which two components may be develop the chemical bond in between. So this kind of chemical who make a bond or break a bond, the procedure is governed by a particular substance, and that is known as an accelerator. And this accelerator move and push away the process on such a direction that process will become faster and faster. So that particular purpose will be associated and fulfilled by the enzymes in the biological reaction. So let us move ahead and discuss various features of the enzyme. So what we think about the enzyme? Enzymes are what? It is a biological catalyst, it is the protein. So as it is a protein, it has a various functions. Okay, and as it is a protein, it has a various features. What kind of feature protein have? Let's discuss. So proteins are basically those components who can work efficiently, who can work and hold their structure efficiently at a particular temperature, and that temperature in our body is about 37 degrees centigrade, right? And that is about 98.6 degree Fahrenheit, which is nothing but a normal temperature of a human body. Okay, which is the normal temperature of a human body. So in this particular scenario, we have a 37 degrees centigrade, which is a temperature on which the human bodies or human enzyme can work most efficiently, and that particular temperature is known as the optimum temperature. Okay, I think you get this point, right? This kind of temperature is known as the optimum temperature. I think you all people get this, yes or no? Okay, yes, right. So the enzymes basically work on the optimum temperature, and that optimum temperature in this particular case is around 37 degrees centigrade, right? But what happens when your body temperature get rise? What happens whenever a person get a fever, right? Fever is a state in which what happen? The body temperature rise from 100 degree Fahrenheit, which is around 39 degree or 38 point something, right? 38.9 degrees centigrade, or it is 39 degrees centigrade. When it exceed beyond this, at the temperature around, you can say more than 40 degrees centigrade, okay, the temperature more than 40 degrees centigrade, the enzymatic structure breakdown. Why and how? Let us discuss and let us understand this. For that purpose, I want to show you and demonstrate you a structure of protein, that how the protein structure is formed. So our topic now little bit deviated, our topic now being little bit diverse on the protein part rather than the enzyme part. Remember that the proteins are basically of four types. It composes four types of configuration. The first configuration is a primary configuration, in which what happens? The amino acid join each other in a straight chain like manner. Okay, so it is a chain, it is a flat chain, and that flat chain is what? It is a primary protein, or you can say it is a primary configuration of protein, right? On the other hand, what happens? When this particular chain will become longer and longer, what they make? they will form the hydrogen bond in between. Why? Because as we learn that it is nothing but the derivative of methane, and one hydrogen is still there. Okay, one hydrogen which were replaced by the amino group, one hydrogen which was replaced by the acid group, one hydrogen was replaced by any random group, according to which the enzyme structure is manipulated and you can identify the enzyme or a protein. In other case, in amino acid, right, the random group is responsible for the variety of amino acid, but in this case one hydrogen is still there. So when amino acid chain will become longer and longer, it will now generally what do? It will do a circular folding, and this circular folding will make it what? The secondary configuration, right? So this is the primary configuration, this is the secondary configuration. Now after that what happened? The tertiary configuration is formed, right? So what is tertiary configuration? When this secondary type of configuration, it combine, or you can say it reticulate in such a manner that enzyme now what? Enzyme now being behave, or you can say protein is look like this. So this kind of protein is known as a tertiary protein. And last but not the least, that is the quaternary protein. Okay, that is the quaternary protein. Quaternary protein is what? I think you people might not get this idea very easily, so now we have to do something different. Okay, so I will demonstrate you how this structure is formed. So wait for a minute. Okay, right. So you might see a paper over here, right? So suppose, let's say this is the paper. Okay, so under this paper, if we say the amino acid is stand like this, see, this is the paper. You don't have to look on the paper, you have to just see and focus on this margin. So this margin compose what? This margin compose one amino acid over here, one over here, third one is here, and fourth one is here. So there are lots of amino acid present in the sequence. Are you getting my point? Right? What happened here? The one amino acid is here, second, third, fourth, and fifth. So you have to just look on this structure, or you can say you have to just look on the paper margin. Clear? So this type of structure is what? Generally this type of structure is known as a primary structure. But when this chain become longer and longer, what happen? The two amino acid bind the hydrogen bond with each other, right? What they do? They do a hydrogen bond, they form a hydrogen bond which is a weak bond. Okay, it is a weak bond in between. So if we show that secondary kind of configuration, it will look like this. See, right, now I have a paper. But this paper is actually what? This paper is actually folded. How and why this folded? Because the one amino acid, second amino acid, maybe the third amino acid bind a hydrogen bond with the first, right? So now it become a zigzag type. Why? Because the length is higher, so attraction will get, and due to that particular attraction, the hydrogen bond will form in between two or successive amino acid, and due to that what happened? The secondary kind of structure will be formed. But when this chain will become more and more longer, some strong bond will be formed, and that strong bonds are what? The sulfur-sulfur bond, one. Another one is, actually this is not sulfur-sulfur bond, this is the sulfur-sulfur bond. Another one is ionic bond, right? So ionic bond, sulfur bond, and so on will form. And due to that what happens? See, if this is the secondary configuration, due to lots and lots of hydrogen bond, due to lots and lots of sulfur disulfide bond you might say, and due to lots of the ionic bond, what happened? The structure will start to collapse, right? The structure will start to collapse, and now it is your tertiary protein. Okay, this is the tertiary configuration of protein. But the interesting fact is, remember this, the primary configuration, what is primary configuration? A newly formed polypeptide chain, right? So the newly formed polypeptide chain, they are normally what? A primary. Okay, they are normally a primary configuration. Please note down this fact because it is not mentioned in NCERT, but examiner can ask you, right? So secondary configuration, where it is found? It is present in the cases such as a keratin, okay, keratin protein, which is a protein of our nails and hair. Okay, so it is present in keratin as well as the fibrin, or we can say fibrinogen, right? So these are what the fibrous proteins, and this fibrous protein mainly compose or mainly organize in such a manner that they shows you the secondary type of configuration. But in the third case, if we talk about the tertiary type of configuration, under tertiary type of configuration, the proteins, they just normally behave like the enzymes. Okay, the example, we see the example, see, where we can find a primary configuration, we find a primary configuration under such a cases where the newly formed polypeptide chain is arise. We find the secondary configuration in such a case where the keratin, fibrinogen, this kind of protein is formed, and we also see the tertiary protein in the case of enzyme. So most of the enzymes, they are the tertiary configurated, clear? But there are some enzymes which are even more complex, which is quaternary configurated. Now what do you mean by quaternary configuration, right? So if I just unfold it for the sake of understanding, because we have to save the tree and the papers are generally formed by the tree, so we will not ruin these trees surround us because it is very important under the ecological convention, right? So here you can see, let's say we have a one tertiary protein here, we have a second tertiary protein over here. This is, you might say, the another and a 4, right? So what happened when a one tertiary protein binds with another, the third join it, fourth join it, or maybe several join together? When more than one tertiary type of protein settle together, join together, then the structure will be formed, and that particular structure is the structure of quaternary protein. But in this case, where we just see the four tertiary chain together, or you can say a complex of 4 tertiary chain, what is the example? Example is the hemoglobin. Okay, not the pure hemoglobin, but the globin of this hemoglobin, right? So hemoglobin composes 2 parts, right? One is the ionized part, which is the heme. The another is the globin part, which is a protein. See, these sessions are normally for your betterment. These sessions are normally for the improvement of yourself, right? So please do not think that the topic was enzyme and now we learn about the protein configuration, because everything is integrated, right? You have to learn, you have to understand each and every fact under this such a manner that you will never ever be compromise yourself to give attention in any topic, right? So this is the structure. Hemoglobin is basically made up of 2 units. One is a heme, second is globin. Under the hemoglobin structure, the heme is only just about 5%, where the globin is 95%. So who is major? Who is dominant? The globin. How globin is formed? So globin compose 4 chain. The hemoglobins, globin compose 4 chain. 2 alpha chain, 2 beta chain. So maybe this 2 is alpha, maybe this 2 is beta. So more than one tertiary protein, you might say that more than one tertiary protein configuration came together and formulate the quaternary protein. So this was the demonstration of the enzymatic structure and enzymatic behavior. Now one more important thing is that every enzymes are protein? No, actually not, right? Not all the enzymes are protein. Actually, the 99.99% enzymes are protein, but there is an exception, and it might be the question of this year's NEET examination, not tomorrow, maybe in the year 2022, right? So please remember this, there is an exception, and the exception is what? That is a ribozyme. Okay, that is the ribozymes which is present and behaved by the rRNA which is present on the ribosome. So ribozyme is a nucleic acid. So whenever examiner asks you a question that which one of the following is correct or incorrect, I share something that in the year 2012 there was a question, year 2012, where the examination was AIPMT, right, All India Pre-Medical Test, in which one particular question was come that which of the following is incorrect. Okay, what they say? Incorrect. So the other three sentences were very complicated, but with these complications, or you can say with this complexity, one particular option was very clear, that shouting that all the enzyme, majority of enzyme, they are made up of protein, but some of them is made up of lipid. No, that is wrong. No lipid molecule behave like an enzyme. There is a molecule such as the ribozyme, which is nothing but the rRNA, okay, rRNA present in ribosome, and that is our ribozyme. Clear? So ribozyme might be, see, the ribozyme might be the enzyme. So 99.99% enzymes, they are protein, but there is exception. Now we might have to see some features of the enzyme. That is the characteristic of protein as well. We learn that majority of enzyme, they are nothing but the tertiary proteins, right? We learn that majority of enzyme, they are the tertiary protein. What tertiary protein is all about? Tertiary protein is the group of amino acid bind with the several bonds such as the peptide bond, which is the very primer bond, right? Which is very prime bond, the peptide bond. Another one is the disulfide bond. Third one is the ionic bond. So there are lots of bonds. Okay, when temperature is rising, we learn that the 38 degrees centigrade or 39 degrees centigrade is the one of the temperature. It is a stabilizing temperature on which the enzyme can proliferate, they do their optimum work, and that's why that particular temperature is known as the optimum temperature. Clear? So that is the optimum temperature. In this case, what happened? When the temperature will fall down less than 38 degrees centigrade, the enzymatic activity will reduce slowly. Why? Because the bond formation will not happen. What is the mechanism behind the bond formation? Why bond has to be formed? We will discuss them later on. But remember, when you go beyond the 42 degrees centigrade, maybe it is greater than 42 degrees centigrade, our body, which is the temperature, it is a hyperpyrexia, right? It is a hyperpyrexia where the body temperature may be exceed from 104 degrees centigrade, which is very high fever. Under this situation, what happened? The bonds in between the amino acid will be breaked, and due to that the structure will break, and that destruction in the structure will lead or will result into what? Will result into the declination of the work efficiency of any enzyme. So this thing might phase you, that whenever you increase the temperature, remember this, whenever you increase the temperature of a particular system in such a manner which is very high degree, then your enzyme will lose their efficiency to work. But the question is how an enzyme works, right? The prime question is how an enzyme is works. So we have to first optimally discuss them, that how an enzyme works, and later on we will discuss the factor affecting on them. So let's just simply discuss. Yes, so how enzyme work? Enzyme work very simply. See, for the discussion of enzyme and its work, we have to first look on the structure. Might say this is particular enzyme. Okay, sir, it is a cell. No, not it is a cell, it is an enzyme. Maybe it is an enzyme E. Okay, let's take any example. It might be the enzyme like the hetero, or you can say hexokinase, right? Hexokinase is the enzyme used for what? The breakdown of any particular machinery, which is the hexo sugar. It might be used in the formation of glucose 6-phosphate from the glucose. Clear? So what happened during this procedure? Focus it, right? So here, if we join and particular substrate, okay, so this is what a substrate. We have to draw this carefully. Okay, this might be the enzyme. Okay, so this is the structure of enzyme, and this is what? This is the substrate. So this is enzyme, this is substrate. Now this is a schematic diagram. Okay, proteins are not like that. It is a tertiary configuration where there are certain points. See, if this is a tertiary configuration, you might say, yes, there is a groove. Okay, there is a groove compose various ability. Okay, there is a groove who compose various ability, right? Like this. See, this compose various ability to do or to proliferate, to promote the chemical reaction. Okay, or it is able to do a enzymatic work. So the whole dependency of enzymatic activity or efficiency of enzyme is depend on this particular site, and this particular site is known as the active site. Okay, but after that it will be lengthier, it will be interestless, right? So we have to draw a schematic diagram, and under this schematic diagram we will discuss them, right? So here is enzyme, here is substrate. What happened? This enzyme and substrate bind together. Okay, this enzyme and substrate bind together. But first thing first, this active site, here in this case, this is the active site. I think you notice something, that the active site, the shape of active site is completely opposite than the shape of what? Shape of this substrate. Remember this. The enzyme work like what? It is work like the template. The active site will work like the template on which what comes? This substrate came. So always remember, the active site is a particular place where the substrate will join. So the shape should be like a lock and key. So here is the mechanism which is very similar to lock and key. And if we say it is a lock and key procedure, you know that lock should be specific for a particular key, and key is specific for the lock. So that's why the enzymes are specific to the substrate, and substrates are also very specific to the enzyme. Remember this, right? I again repeat. See, the enzymes are the lock and substrates are the keys, or maybe you can say vice versa, that the substrate is the lock which wants something to be happen, and the enzyme is the key, or you can say the active site of enzyme is the key for that particular reaction by which the reaction will start and proceeds. So what happened here? That enzyme, this enzyme binds with what? Enzyme, or you can say active site binds with what? Active site binds with the substrate. Maybe this is the substrate. In this case, what we use? We use a hexokinase. Hexokinase is the enzyme responsible for the formation of glucose 6-phosphate from the glucose itself. So what it do? It will now convert this particular substrate at this particular point. See, you all know that enzymes having a particular property that they will never ever participate in the process. Okay, enzyme never participate in the process. Enzyme just proliferate or accelerate the reaction. So enzyme will never change, enzyme will never ever do anything under this reaction. It will just promote it towards the productive side. So see what happened? When substrate join with the enzyme and substrate will lead to the formation of some bond, and that is known as the transition stage. That is known as what? Transition or transitional state. Okay, so what is transitional state? Transitional state is a particular state in which what happened, or you can say a phase of time frame, right, in which what happened, under which the enzyme and substrate, they form a new bond and break the previous one. Okay, they form a new bond and break the previous one. Okay, so under this what happened? under the enzyme, now the product will be change its nature, and now product is converted into, sorry, you can say, sorry, my mistake, here is a substrate, now substrate is convert into the product. So what happened? The reaction is such like this: enzyme bind with the substrate, enzyme binds the substrate, and what happened? The formation of enzyme substrate complex. Okay, so this is the enzyme substrate complex. Afterward, what happened? And in between the enzyme as well as the substrate, new bond form, older bond will break, and it convert into a transitional phase, that is the EP, enzyme product complex. Okay, that is the enzyme product complex. And afterward, what happened? The enzyme is released, enzyme is now released, and the product is released. So product change the, or you can say substrate for change their state, and it is now become the product. Is it clear? So this is whole idea, whole crush of the enzymatic activity. So enzymes are working like this, right? So this is what the procedure. So enzyme plus phosphate, enzyme plus the product is formed, and now product will do certain function. So this is the way how enzymatic activity is done. But remember, enzymes are very fast as well as accurate. Okay, enzymes are very fast as well as accurate because the enzyme are specific. One particular enzyme work for one particular substrate, and a particular substrate can be catalyzed by only a specific type of enzyme, because the enzyme and substrate relation is like the lock and key relation. So let us look further. As we say that enzymes and product, the process of substrate to product is very fast, very quick. How much it is quick, right? In the presence of enzyme and in the absence of enzyme, that we have to discuss over here, right? So first of all, I have to watch some of the comment. Okay, Vijay Rupani resign, right? We don't have to do deal with the Vijay Rupani at all, right? Deep, yes. Okay, so please guys tell me, are you understand all the facts, right? If there is any doubt, please make sure to tell me in the later phase of the session, or you can say in the last of the session. Okay, okay, yes, please focus on this study, right? Thank you Tanisha, thank you Twisha, thank you Rajveer. Okay, let us discuss furthermore. Now we say that enzyme make acceleration under the process. What happen if enzyme will not participate in a reaction, and the reaction will through the without enzyme condition? Let us see. If we take a case of carbonic anhydrase, you might know about the carbonic anhydrase. Carbonic anhydrase is what? Carbonic anhydrase is a particular enzyme used in this reaction, in which what happen? Water binds with, or you can say water react with the CO2, and it form the H2CO3, which is carbonic acid. Okay, which is carbonic acid. Remember, this process is not governed only by the carbonic anhydrase. Actually, this process want what? The support, and that support is basically came from the Zn+2 ion. Okay, so zinc is the ion, zinc is the cofactor, right? Zinc is the cofactor, or you can say coenzyme for the carbonic anhydrase. Without zinc, carbonic anhydrase is just merely a protein molecule who cannot do anything, right? So if carbonic anhydrase has to work, if you want to join the H2O and CO2, you have to use the zinc in this particular process. So zinc is what? It is a cofactor, it is a coenzyme as well. Now you might say there is some reactions like this. This is the reversible reaction, and it is also done outside the body, right? Enzymes are work under the body. This kind of process is done outside the body, and you will be surprised that majority of CO2 on this particular world in environment, majority of this CO2 is dissolved actually inside the water in the form of carbonic acid. Clear? Maximum amount of CO2 where it is in the water. How this reaction takes place? There is no enzyme. So yes, reaction can be done. Without enzyme it can be done. But the speed of this particular process will be very, very, very slow. Right? The speed of this particular process will be very slow. What is the speed? Let's say this particular procedure which is without, you can say it is without the enzyme. Okay, it is without enzyme, and the speed is what? Speed is around 200 molecule. You will get 200 molecule of carbonic acid from 200 molecule of H2O and 200 molecule of CO2 every hour. Okay, so if you put a sustainable amount of CO2 with a sustainable or equal amount of H2O, there will be only 200 molecules which is reacted and formulated. So only 200 molecules of H2O bind with the CO2 without the enzyme. But in the presence of enzyme, this reaction is something different. This reaction will be accelerated in such a direction that this reaction with carbonic anhydrase with enzyme result into around 6 lakh molecule every second. Okay, right, there is 600 you can say 6 lakh molecule every second. So what enzyme do? Enzyme merely do nothing, but it is massively done a change in the speed of reaction. It is not manipulated, it is not changeable towards the reaction. It is just a parameter which change the speed of enzymatic would change the speed of reaction itself. And how much this regulation occur? Here you can see the process is faster up to the 10 you can say 10 millions even. Okay, 2 to 10 million time, right? So this reaction is merely very, very fast. Okay, it is very, very fast. So this is the whole idea about the enzymatic activity and its efficiency. You might say that we need the enzyme, this particular enzyme is this much faster, but everyone will not. No, our body everly do the glycolysis, maybe you can say. Glycolysis is what you know about the glycolysis. Glycolysis is the reaction in which glucose is breaked under 2 molecules of pyruvic acid, and this particular reaction needs 10 steps, and this 10 step having 10 enzymes. Okay, right? So the reaction itself needed 10 enzymes, and this reaction should be faster. What happen if a dog chases you? What happen if you will come under a various situation, such a situation in which the life is in the threat? What you have to do? You have to run. For the running, what you have to break? You have to break a glucose for the energy, and by this energy only and only you can survive. Clear? So these are the enzymes who makes our life utilizable. So before enzyme, in your NCERT, in the chapter number 9 of 11th NCERT, they gives you a topic, that is the living state, and they say that a living state is merely nothing but the unstable state of a living being. Okay, it is a instability under stability. Okay, right, it is a stability under instability. So this is the core of the biological processes, right? So biological process can only and only be done with the help of enzyme. So we might see the whole idea of enzyme, its activity, its nature, its composition, its structure, everything. Now we have to discuss the factor affecting on the enzyme. But prior to that, I want to ask you that, is there any doubt, please? Is there any doubt, please? I'm just waiting because the screen is actually lagging, right? The streaming is actually lagging about 15 to 20 seconds. So I have to wait for your questions. Yes. If there is no question, please tell me no at least. And please don't forget to like, share and subscribe these videos, right? And we also, yes, good. If you don't have, crystal clear. Thank you, Akanksha. Thank you so much. Thank you very much. Yes. Okay. Okay, so if you are new on this channel, you can watch the previous session as well, and this type of session will going in on this channel, particularly in two medium. One is the English and one is the Gujarati. Unfortunately, our national language, which is the Hindi, is not available on this channel, but I will do, on the request of some of the student, maybe the next lecture or next session will be in the Hindi language. Okay, right, the content will be English. Yes, I don't know about the Hindi content actually, that's why. Okay, so let's move ahead. Okay, so next topic is what? The next topic is the factor affecting on the enzyme. What kind of factor they are affect the procedure? What kind of factor they affect the enzymatic activity? So we have to learn about the three factors particularly. Okay, what those three factors? The first factor is the temperature. The first one is temperature. The second one is pH. The third procedure that we have to discuss is the concentration of substrate. Okay, so we have to learn about the temperature, what enzyme do, how enzymatic activity fluctuate due to the change of temperature, how enzymatic activity will speed up, or you can say it decline with the change in the pH, as well as we will discuss about the next topic and the last topic, which is the concentration of a substrate, right? So let us first move on the temperature basis. So how temperature affects? Temperatures affect normally like this manner. See, it is actually right for both, for the procedure of the temperature or reaction under the temperature as well as pH. Okay, so here what we put the enzymatic activity. Here is the enzymatic activity, and maybe here is the temperature. The graph is like this. What happened here? We know about the optimum temperature. We discuss about the optimum temperature. Optimum temperature is that temperature on which enzyme shows a maximum value of the reaction. Okay, it shows a maximum value. It shows the maximum activity, and that temperature is about, you can say, 38 degree centigrade, or it might be 39 degree centigrade. And we learn that as enzymatic activities decline, now you are in the position, yes, now you are in the such of a position in which you can give a little classical, you can say little developed answer for this question, that why the enzymatic activity goes down below the 38 degree centigrade, or you might say below optimum temperature. Remember that optimum temperature is which, where the enzymatic activity is faster. Why? Because the older bond will form very easily, right? The older bond will form very easily. So we don't have actually the figure which we drew right before. So basically what happened under the enzyme, okay, in between the enzyme and substrate, there is a formation of bonds, right, and the breakdown of the prior bond and a formation of new bond. This particular reaction will be work efficiently on 38 degrees centigrade, and you know that chemical reaction will be rise up as the temperature goes up. Why? Because as the temperature rise, you can say on the very temperature 0 degree centigrade, what is the kinetic energy? Kinetic energy is about 273 Kelvin. Okay, 273 Kelvin. On the other hand, you can say at the room temperature, which is around to be 27, 25, and it is around 298 Kelvin, or you might say it is a 300 Kelvin if it is a 25 degrees centigrade, right? So what happened? As you increase the temperature, the kinetic energy will be increased. And what is kinetic energy? Kinetic energy is the energy in which the molecule will move. Okay, it is a mobile energy, or it is the energy on which molecule can move from one side to another side in the system. So under system, if you want to move a particular particle, if you want to move a particular substance, what you have to give? You have to give a sufficient amount of kinetic energy. So when we give the kinetic energy which is sufficient, is what? It is around 318 degree, 318 Kelvin, right? Maybe it is more higher. So 38 plus, you can say 273, what it is? It is something around 313 Kelvin, right? So 313 Kelvin is the kinetic energy on which it do a optimum work. So when you reduce down the optimum, and you can say you reduce down the temperature, you reduce down actually kinetic energy. So this is whole play field of a kinetic energy rather than a temperature. So when you reduce temperature, ultimately there is a fall in a kinetic energy, and when there is a fall in a kinetic energy, you might say the bond will not form that efficiently, or bond will not break that much faster. Clear? So that's why enzymatic activity goes down as what happened? As the temperature declining, right? And it is around 12 to 15, it is around 12 to 15 degrees centigrade where enzymatic activity is near about zero, right? It is near about zero. Now you might get a question, sir, as you say that as you rise the temperature, the kinetic energy is rising at the 3 and 13, okay, 330 Kelvin, there is some kinetic energy. But we increase this temperature, what happened? When we increase this temperature, you might say up to 318 Kelvin, okay, 318 Kelvin, kinetic energy more increase. And if kinetic energy and enzymatic activity is proportionate to each other, this reaction should be increased. The speed of reaction or efficiency of enzyme should be increased. But why it is declined down? Okay, why it is declined? Because of the structural deformity. As we learn that in a tertiary structure, this is the tertiary structure of protein, okay, tertiary structure of protein, which is the enzyme. When you increase the energy, when you increase too much of energy, there is a breakdown of disulfide bond. You learn about the disulfide bond like this. Okay, so these are the disulfide bond. So when there is increase in the kinetic energy, on the other hand, there will be what? There will be breakdown in such kind of bonds, such as the hydrogen bond, such as the disulfide bond, such as some ionic bond, or maybe there will be the formation of new bond. So ultimately it will denaturate, it will change the shape of active site. Are you getting my point? When the enzyme structure will change, active site will change, and when active site will change, what happens? The enzyme will be not effective, because it's just like a lock and key, right? So lock, if you manipulate just a little under the lock, what happens? Key will never ever able to open that. Clear? So that's why when you increase the temperature more than 40 degrees centigrade, you can say more than 42, 45 degrees centigrade, what happens? Just there will be more and more kinetic energy, and under which what happens? The bonds will break, the structure will denaturate. So what happened? At the very high temperature, around 45 degrees centigrade, the enzyme, on the other hand, it is a protein, they break. Okay, and what is the term we use for the breakdown of this kind of enzyme or protein? It is a denaturation. Denaturation. So you have to remember, if examiner asks you that why enzymatic activity goes on the back foot, or you can say decline, as we increase temperature after optimum pH? And answer is very simple, because of the denaturation of protein. Okay, protein gets denaturated, and due to that what happen? Enzymes are braked, enzymatic activity will also reduce. So this is whole concept by which we have to go through and by which we learn. Is it clear? So this is what the idea of enzyme on the basis of temperature. But what about the next topic, and that is the pH? How it behave in the influence of pH? Maybe here same, the activity of enzyme, and on other hand we have a pH. So there is a 7 pH, our blood pH is around 7.4. Here is 8, 9, 10, 11. So we move up towards the alkaline medium. Here is 6, 5, 4, 3, 2, 1 and 0. So normally majority of enzyme, remember I use the term majority, it is also applied on the temperature. Every enzyme does not having the optimum pH which is a 38 degrees centigrade. It will be reflecting, or you can say it will be changeable, right? So majority of enzyme, they are not worked after, you can say, around the 2 pH, or which is very acidic, okay, or below the pH which is 2. And majority of enzyme that does not work after the 11, okay, that does not work after 11. Here you can see this is the pH, this is the pH where the enzyme do a maximum work. So that is pH known, that pH is known as the optimal pH, as like the optimal temperature. Now you might get a question, sir, what about lysosome? We'll talk about the lysosome. That a lysosome normally compose a particular group of enzyme, and it is about 50 type of enzyme, right? The lysosome compose about 50 type of enzyme, and this 50 type of enzyme, they are acid hydrolase. That means they do a perfect optimal work at the low pH, and majority of you might know about the pH. Please write down, right? Please write down the optimal pH for the substances or enzymes like a lysosomal enzyme. Okay, right? So I will wait for that particular, that you will give right answer. So the pH is what? pH is around 5. At the 5 pH, the lysosomal enzyme gives the maximum work. Then this phenomena will change. But we say that it is for a normal situation, normal circumstances. Majority of enzyme do this. But there are some enzymes who activate and behave their optimum function on the low pH. Some of them will activate and behave at a high pH. Like what? Like the enteric enzyme. Okay, enteric enzyme. So what is enteric enzyme? Enzyme under intestine. Okay, enzyme in intestine. The intestinal enzymes such as the pancreatic enzyme, right? The trypsinogen, chymotrypsinogen, procarboxypeptidase, these all are the enzymes who work at a very high pH, around 8 or 9, because they are the alkaline enzyme. Okay, so this phenomena will change. Okay, this phenomena will change, right? So temperature also behaves like this. Temperature also behave like this. Majority of enzyme, see, majority of enzyme, they denaturate, the protein breaks at the temperature which is around 42, 45, 50 degrees centigrade, right, or more than an optimal temperature. But there are some bacteria which are the Archy bacteria, and you learn about the Archy bacteria that normally the Archy bacteria grow, proliferate themselves under the situation or circumstances which is very in the high temperature or high salinity. Clear? Let's talk about the Archy bacteria. One of them is thermoacidophile. Clear? So thermoacidophile, and one enzyme, one bacteria which is a Thermus aquaticus. Okay, so Thermus aquaticus is the bacteria by which we get an enzyme, that is a Taq polymerase. Okay, and that is very important question, very IMP question of standard 12, chapter number 11. Okay, chapter 11, right? So tag polymerase is used in the procedure such as the PCR, polymerase chain reaction. Okay, polymerase chain reaction. You learn about, you maybe know, you maybe heard about the RT-PCR, which is a reverse transcription polymerase chain reaction. Okay, RT-PCRs, which is a test usually done for the COVID-19. Okay, for the diagnosis of COVID-19 or coronavirus. So that PCR processes are done at a very high temperature on which there is enzyme work is a tag polymerase. So these are the enzyme who can survive, who can work efficiently at 80 degrees centigrade or 90 degrees centigrade, which is way beyond the temperature on which our enzyme is work. Clear? So our enzyme normally doing a well function at optimum temperature, right, which is around 38, 39, but there are some enzyme who can do the function work at around 80, 90 degrees centigrade. And what are those organisms? They are just basically Archy Bacteria. So the Archy Bacteria is like a thermoacidophile. Their enzyme will behave and their enzyme will efficiently work on this particular temperature as well. So let's move ahead and discuss about another one, and that is the concentration. Okay, let's talk about the concentration of substrate. Okay, and there is evidence that as we grow, as we rise the level or concentration of substrate, enzymatic activity is also increases. How? See, if this is a figure or if this is a graph, here is the activity of enzyme and here is the substrate level. What will be the process? Okay, is graph is similar with pH? No. Okay, graph is not similar with the pH as well as temperature, but graph is normally shows like this. Okay, and here there is a saturated phase. How this is achieved and how it is works, let's understand. When the substrate level is 0, right, what we learn that the enzymatic reaction is enzyme plus substrate. It forms the enzyme substrate complex. This enzyme substrate complex goes under the various changes, the intermediate changes, and it is now converted into the enzyme product complex, and afterward enzyme product complex dissolve, and the dissolution will give result into the product. Okay, the dissolution gives result into the product. So we get a product, right? But where you get a product? When you have a substrate. If there is no substrate, then how anybody can expect the product, right? So enzymatic activity will obviously, the product level will obviously 0. Okay, the product level, you can say the achieved product will be 0. As you increase the level of substrate, right, you put a 5 molecule of substrate, 10 molecule of substrate, initially enzymatic activity will grow rapidly, right? The enzymatic activity will grow rapidly. How and why? See, right, this kind of activity we can see, and this kind of activity we can found in the normal situation, normal circumstances. How and why? Let us understand. If I say that maybe this is just a hypothetical situation to understand the phenomena and to understand this graph, right? If I say you have X amount, you have a X amount of enzyme, or you can say X amount of substrate. Let's take X as the 300 molecule, right? And we have 300 enzyme as well. Okay, at that time we have 300 enzyme as well, and one enzyme, remember, the one enzyme is capable to react with the 3 substrate at a time. Okay, 3 substrate at a time means what? That one particular substrate need about the 0, or you can say 33% of the second. Okay, how and why? Because the enzyme, one enzyme is capable to convert 3 substrate into the product one substrate will take around 33% part of, you can say 0.33 second, 0.33 second to convert into the product. So what happened here? When you put a 300 molecule, is the 300 molecule will govern by the 100 molecule of enzyme? Yes, right, because one enzyme will take how many? 3 at a time. Okay, so then how the enzymatic activities increase? But that is not a case. What happened? That if we have a 300 molecule, yes, there is a capacity that in 1 second enzyme can convert 3 substrate, but it will not perform. Instead of that, every particular, each and every enzyme will take how many? 1. So reaction will be completed within a few seconds. Okay, within a fraction of second, you might say the whole reaction in this case will finish under 0.33 second. I hope you might understand this. It is little bit mathematical, and as you are the biological student, right, biology student may be or may not be dislike the maths or mathematical formula, but please understand this carefully. If you don't get, please go back and listen this carefully. So let's take another example. If I take a blank slide, see, this is the blank slide on which we have to perform. Now if I take a 600 molecule, okay, now if I take a 600 molecule, what happened? Right, under the first circumstances, what happened? This reaction can be done in the 0.33 second, okay, but it will not, because it will take a 1 second, because when you add a 3 substrate with the enzyme or a 1 substrate with the enzyme, no difference will occur. Enzyme clear this reaction at a set time, because it may have the 3 active site. If they have a 3 active site, 3 active site will work simultaneously. Okay, so it might get around 1 second to perform this particular reaction, 1 second for 300 molecule. So the process is little bit, you can see here. After that, if you increase the 600 molecule, okay, if you increase the substrate level to the 600 molecule, how many seconds they get? Okay, how many seconds they take? Now the one enzyme fill what? How many of active site? 2 active site. One enzyme may fill 2 active site. So 2 active site work simultaneously, but they can work with the 3 active site. So no matter one active site is blank, the work will done at the same time. So it is also take a 1 second. These are the hypothetical numbers, right? You don't have to go with this number. These are just fractions of hypothetical situation. Now if you improve or increase the substrate level, because we talk about what? We talk about the substrate level. If you increase the substrate level up to the 900 molecule, we have a 300 molecule of what? Enzyme. We have a 300 molecule of enzyme, and we have a 900 molecule of substrate. Then what happened? That one enzyme, they will filled with the 3 active site. So all those 3 active sites are now in the work. Okay, all those 300 active sites are 300 molecule of enzyme. They're all each 3 active site will be now interested and involving inside the activity. So in 1 second, right, in 1 second they will give 900 molecules. Okay, 900 molecules. This is all right. Now what you do? Here there should be the 300 molecule substrate level. Here there should be the 600 molecule substrate level, and here there should be 900 molecule of substrate. But when you increase it up to the 1200 molecule substrate, activity will never change. Why and how? See, right, if we take a 1200 molecule, how many of enzyme it require? 400, because 400 into 3 is equal to 1200, but we have only 300 molecule, and those 300 molecule have a maximum capacity to hold 3 active site at a time. So at a 1 second, what happened? 900 molecule will be added. Okay, 900 molecule will be added. So activity will be stay at the place as it was on the 900 molecule concentration. So activity will now never ever increase. So that is what, that is a saturation, that is known as a saturation of enzyme. Okay, that is the enzymatic saturation. Now enzyme will never do beyond this limit. Are you getting my point? Right. Now you might say, sir, at the 1200, at the very first second, 900 molecule will be proceed into the product, but what about the remaining 300 molecule? But here we see that 1200 molecule every second, at very next second you have a 1200 molecule again. So now you increase as much as you want this concentration of substrate, the activity will remain stagnant, the activity will remain static. Okay, so that is the beautiful idea about the enzyme. So this is, you see, this is the activity chart of the enzyme. We already discussed about them. The next topic we have to discuss is the enzymatic reaction. We already discussed, but the major question is, we learn that enzyme such as a carbonic anhydrase, they can produce the such effect under which the procedure will rise up, and it is rising up about 10 million times. It is very huge number. Okay, 1 crore time or 10 million time. How this much of acceleration takes place? And that is our process of interest. Okay, right? So, sorry. So what happened here? Let's understand. If I draw a graph, when a substrate convert into product, they have an energy difference. Okay, they have an energy difference. Energy difference is what? If you take a process such as the endothermic process, right? Endothermic means in which you have to put the energy, right? In which you have to put the energy on, in which you have to give energy. So their energy level, the product energy level is higher because you have to give energy. Okay, you have to give energy. So the product energy level is higher. But normally enzymatic activities are exothermic in nature. Exothermic in nature, and you might learn exothermic. Okay, and you might learn in chemistry, exothermic reactions are those reactions in which what happen, energy is liberated instead of used. Two type of reaction is there. One is the endothermic, second one is the exothermic. Endothermic reactions are those reactions in which what happen, that the energy is conceived, energy is consumed by the substrate to convert into product. And on the other hand, the exothermic reaction is what? The such a reaction in which we have to give energy, otherwise reaction will not take place. So in this case we have to give energy, on the other case we have to in the exothermic energy will release, right? Energy will liberate. So obviously if substrate release energy, the level of energy, see, level of energy, I repeat, you see, I don't want to take any risk because you might not understand. If you learn carefully in the chemistry, the enzymatic activity or any kind of reaction, chemical reaction, they have a two type. The first type is the endothermic reaction. What do you mean by endothermic reaction? Endothermic reaction means in which you have to put, in which you have to give the energy from outside to convert into the product. So product just get that energy and it takes the energy. Okay, that reaction energy they get. So energy level will be higher in product and lower in the substrate. But in this case, in enzymatic activity, majority of them are the exothermic reaction. And under exothermic reaction, chemistry law says that the exothermic reactions are those reactions in which the energy will liberated, energy is released from the product. So when substrate convert into product release the energy. So you may say the product, the substrate X is converted into the product Y. Who has more energy? Obviously X, because Y release energy, and when Y release energy, energy conservation law will come, and energy conservation law says there is no formation, no deletion of energy. There is only and only transformation. So the exothermic energy, the exothermic reaction says what? That during the procedure, product form and heat is released, and heat is nothing but a kind of energy. So energy level of substrate is high than the energy level of product. So energy level of product is basically what? The energy level of product is low in the compare of substrate. I hope you might understand this particular thing. Yes or no? Right, please give answer. I'm staying here for few seconds, right, because I have to verify that you people at least understand what I'm talking about. Yes. Come on, fast. Okay, fine, fine, Mehak Vedahi. Okay, so let's move ahead. Okay, now you understand that substrate has more energy than the product in exothermic reaction, and majority of enzymatic activities, they are basically what? Exothermic in nature. So we might say that here is the graph. The energy of substrate is little here, or let's just draw little lower. Okay, so enzymatic energy, the substrate energy is maybe at this place. So substrate is normally has to give energy, because for the reaction we have to give energy. For the reaction we have to give a little amount of energy. Are you getting my point? We have to give a little amount of energy. So when we give energy, they take a energy, process is now at that place where enzyme or substrate, enzyme substrate complex is formed. See, at this place what happened? Enzyme and substrate, they are joined. Okay, enzyme and substrate, they are starting to bind, starting to react. Okay, and at that point, in this point, what happened? Enzyme substrate complex is formed. So what happened now? Energy will slowly reduce down, energy will slowly liberating or releasing, and ultimately energy level will fall down. Okay, energy level will fall down way beyond the energy level of the substrate, and product will be achieved. Okay, so product will achieved by what? By releasing of this energy. So first of all, what we do? First of all, we give energy to formation, for the formation of enzyme substrate complex, under which what happen? There is a transitional state. Okay, transitional state. And initial of this lecture, we understand what is transitional state. Transitional state is that particular state on which what happen? Enzyme substrate complex basically convert into the enzyme product complex, and ultimately reaction is done. Right? So we need this much of energy. First of all, we need this much of energy to make enzyme substrate complex, and this particular energy is known as the activation energy. That is known as activation energy. Very important question according to Neet, right? They normally ask this kind of question. What is they show a figure and ask you the A, B, C and other alphabet. So this particular line shows us what? This particular range shows us the activation energy on which enzyme substrate complex formed, and now activation of transition will happen. Clear? So this is the transitional state start after giving this much of energy, which is known as the activation energy. But what enzyme do? This is the activation energy without enzyme. Okay, this is also important. This is the activation energy without enzyme. So we do the reaction without enzyme here. But when we do a process with the help of enzyme, what happened? The activation energy will become down. Okay, enzymatic energy will become reduced. So what happened with the enzyme? You can say this is the reaction start and the product is achieved. Okay, product is achieved. So here also you can say the energy level. Okay, here also you see the energy level, but this energy level is also activation energy. Activation energy. But this activation energy is with what? With enzyme. So this particular stage is what the activation energy, this particular thing says. The activation energy in the presence of enzyme is way down than the activation energy without the enzyme. So this is the core process or core conduct of our study. Clear? How the enzyme reduce the activation energy and how this particular enzyme gives the acceleration effect. Now let's take an example, very hypothetical example. If I need the activation energy, suppose this is the activation energy which is around 10,000 or 1 lakh kilojoule. Okay, this is the energy. If I give 1 lakh kilojoule energy to particular molecule, what happen? They will convert into the product. They need 1 lakh kilojoule as their activation energy without enzyme. Okay, without enzyme. But let's say under which when we use any particular enzyme, the activation energy is reduced from 1 lakh to 100, or you can say even let's say 10 kilojoule. The activation energy is lower down up to the 10 kilojoule. So now you have to give 10 kilojoule energy to convert one molecule into the product, but the energy presence under the system is static. Okay, energy value is static under the system. So when you give 1 lakh kilojoule in absence of enzyme, one molecule will be converted. But in this case, when you give 1 lakh kilojoule in the presence of enzyme, you will see that a 10,000, right, 10,000 molecule will now convert into the product. So the 10,000 substrate now convert into the product. So that's why we can say as the energy level down, the enzymatic activities grows. So enzymatic activity is all based on only one phenomena, and that is what? That is the phenomena of activation energy. As you reduce down the activation energy, the process will be accelerate. So here we finish our fundamental ideas as well as the discussion related with the enzyme. So you can see by reducing activation energy, how it accelerate the process by reduce down the energy. So this was all about the enzyme. Here we finish with the enzyme, which was the first topic of us and which was a very broad large topic of us, right? So this is, yes, the whole lecture is like chemistry. No, not, it is not a chemistry, but it is, yes, it is a biochemistry. So as per your request, now we will shift from chemistry to the real biology or cellular biology. But before that, I want to ensure that everyone should get this particular idea of enzyme crystal clear. Please write down in the comment section, please. Then we will go for the cytoskeleton. Come on fast. Okay, Deep. Okay, Meet. Okay guys, right. So now I get the answer. Now I get the answer. It is little bit lagging, but ultimately I get answer, right? So the second topic that we have to discuss, and it is very easy, right? I will take just only around 15 to 20 minutes to clear both the topics, because both the topics are very small in the nature. Okay, so cytoskeleton. Let us talk about the cytoskeleton. The word cyto means what? Cyto means cell. The skeleton you know about. The skeleton is the holding part. Skeleton is the holding part. So the holding part of cell is known as the cytoskeleton. Okay, clear? Right? The holding part of this cytoskeleton, yes, a cell is known as the cytoskeleton. How and why it is needed? Let's say this is the cell. Let's say this is the cell. Cell compose what? Cell compose various organelles. It might be the nucleus. The nucleus is generally continuated as a what? Endoplasmic reticulum, and ultimately it is convert into the Golgi apparatus, and afterward it will produce various vacuoles such as the lysosomes and all. Clear? Here should be the mitochondria if it is an animal cell, and if it is the plant cell, that there should be the mitochondria as well as the chloroplast. Okay, chloroplast. So these are the components, these are the organelles present inside the cell body. What happens when we put this cell in a static position? Okay, if we take this particular cell in such a direction, what should be happen? The all organelles should be settled down. Okay, all organelles should be settled. Yes, it is just like a fool like things, right? This is a fool like thing, but it is possible. It is possible, yes, but it is not possible in reality. It is possible just in the hypothetical situation or our dilemmas, but it will never ever be done in the real mechanism. Why? Because there is something who just take this all the particle in the position, and that is nothing but the net of protein. Net of protein formulated by what? Net of protein which is formulated by the rough endoplasmic reticulum, right? They are formulated by rough endoplasmic reticulum. Okay, and there are three type of fibers, protein fiber, who hold this whole structure. Okay, who maintain the shape of cell, who gives the mechanical support to the cell, right? And there are three major elements, the three fiber we see here, and the one of them is the microtubule. Remember, the microtubule are the cytoskeletic fibers which compose of or which made up of a tubulin protein, which is made up of what kind of protein? The tubulin protein. And the size of this particular tubulin fiber is around 20 nanometer. Okay, the size of this is around 20 nanometer. The another one is the intermediate, but we have to first talk about the microfilament. See, microtubule you already see in lots of places, just like the meiosis, just like the mitosis. During cell division, what happened? During cell division, there will be the formation of spindle fiber, and you learn that a spindle fiber, basically the spindle fiber is composed of what? Spindle fiber is made up of the tubulin protein, nothing but the microtubule. So microtubule is an essential substance by which what happened, that the particular substance is formed. Okay, right, this is formed. Then microfilament, these are also used in the cell division reaction when the cytokinesis takes place. So during the cytokinesis, what happen? The microfilament will be arranged on the equator of cell. okay, at the equator of cell, right? And here the protein is the actin, okay, the actin protein should be there, and the filament is about 7 nanometer. So these are normally found filament like the microtubule as well as the microfilament, okay. So these two components are broadly seen under cell, but there is rarely seen intermediate filament or intermediate tubules. So intermediate filament or intermediate tubule is basically made up of both the protein actin as well as the tubulin, and size is interbetween. See, here is 7 nanometer, here is 20 nanometer, and its size is about 14 nanometer. So you can write down this information. This is all regarding with your cytoskeleton. So we also finished the second topic, which is the cytoskeleton. Okay. Yes, Elon Musk. What I have to repeat, right? It is very proudful thing that Elon Musk is also present in our class. Okay. Please change your name. I think it is Deep, right? Okay. Right. So let us understand about the thing which is the Kern plus theory. Okay, this is the last topic, and afterward we will finish this session. So Kern plus theory is what? Kern plus theory is the theory given by the Hartwig. This was the theory given by the Hartwig. And according to this theory, what happened? First of all, one question is there: why to study and what is it? Why to study? Because of understanding purpose. The chances of question will be very less, but by this you can understand why cell division is happen. Okay, why cell division is happen? Cell division is happen only and only due to what? Due to the mismanagement in cytoplasmic ratio. What do you mean by cytoplasmic ratio or cytoplasmic nuclear ratio? In the normal structure, in common structure, we have two things. In common structure, we have two things. We have a cell. Cell basically composed of what? Cell basically which is composed of the nucleus. Cells are basically composed of nucleus and cell membrane. So we have a cell membrane as well as nucleus. Now the nucleus is the governor of cell. Who say this? The Hammerling. Remember this. The Hammerling, scientist Hammerling, who perform a various experiment on the algae, which is the unicellular, the largest unicellular alga, and that is the Acetabularia. So they perform experiment on the largest unicellular algae, which is the Acetabularia, and give us what? And they give us the conclusion that nucleus is the governor. Nucleus is the head center of all cell who regulate everything. Okay, who regulate each and every processes. So what happened? During the G1 phase, cell size will be increased. Cell size will be increased. But nucleus size is still that. Okay, nucleus size will never ever increase. So nucleus size is still there and the cytoplasmic volume is increasing. Now the work of nucleus is like antenna. If you put a particular Wi-Fi connection at this particular place, it might access in a certain direction, or you can say in the certain radius. After we go beyond that radius, the Wi-Fi connection will be lost. Wi-Fi connection will ever be lost. So what does this mean? That the nucleus can regulate a particular cell in such a range. Afterward, after you cross the range, everything will be over. Okay, after you cross the range, everything will be over. Okay, so this is what, where the cytoplasmic volume is increased, and afterward when growth will be exaggerated, the cytoplasmic volume, now it will goes over and above than the actual range of the nucleus, right? The range under which nucleus regulate the cytoplasmic affairs, it is now crossed. Clear? So first of all, nucleus is what? It is a 2n. In human being, 46 chromosome, 23 pair, and it composed 2C of DNA. 2C is the value of DNA. Here is also 2C value of DNA. Here is 2C value of DNA. Prior to this stage, Prior to this stage, in this 2 stage, 2C value of DNA was sufficiently able to regulate, to hold, to maintain the cell. But in this particular case, now cell lose their capacity, nucleus lose their capacity to regulate the affairs of cell. So what it do? Now we have to start off the S phase. You know about the cell cycle, right? Cell cycle, basically it composes 3 phases, just like a G1 phase, another one is S phase, another one is G2 phase, and last but not the least, that is M phase. Okay, last but not the least, that is M phase. Clear? So G1 phase, maybe say this is the newly formed cell, just like this. It grow and grow and grow and grow. Maybe it reach over here, maybe it reach over here, right? If cell will reduce down its metabolic activity, or you can say it will balance the metabolic activity, that anabolism is equal to catabolism, formation is equal to the breakdown, cell will stuck here, and that is called as a Z0 phase. Yes or no? That is called as a Z0 phase. But when it reach at that such a point where cell nucleus will not capable, not able even to hold the control, then what we have to do? We have to double the DNA, because 2C value of DNA is not capable, 2C value of DNA is not capable to regulate the affairs of cell. What we have to do? We have to double the DNA. We have to make sure the DNA will now double into 2C to 4C. Where it is done? In the S phase. So when the cell, okay, one very important question was asked in the year 2016, sorry, my mistake, it is 2013 AIPMT examination, as well as in the some of the year of RJPMT examination. The question was like this, that a particular cell enter in blank of the phase, then that has to complete a cell division, and they are bound to be the divide. Okay, so what they ask, whenever a cell reach at the particular point of a cell cycle, then there be no any chances of other pathway. They have to finish the cell cycle, and they have to finish the division, otherwise they can stuck under the Z0, and the answer is S phase. When they enter into the S phase, the DNA volume will become 2C to 4C, and that is the reason now they have to finish the division. Okay, so that is all about the Kant-Class theory. Now you may ask the question, why we have to ensure that the 2C to 4C DNA is done, we learn, right? Because the 2C DNA is not capable to hold the position. But why 4C DNA lead it to the division? Because there is a rule. Okay, there is a rule that n value of DNA, n number of chromosome can only be consist C value of DNA, which makes it healthy, or you can say it makes it the stable. Okay, it makes it stable. But when you cross this, means when you cross this, now see, the chromosome number is still 2n and the value of DNA is 4C. So it is n is equal to 2C. N number of chromosome compose 2C value of DNA. So it is the violation of this n is equal to C, and that's why we have to divide them in such a manner that the number of chromosome will remain same, but the value of DNA will be 2C from 4C. So this is the theory which explains that why cell division is performed and why cell division is happened. So this is all about this, right? Thank you very much for listening me. Thank you very much for watching this video, right? And I hope that you all people get something which is helpful for your preparation. So now I want to ask, now I have to ask little questions from you people, right? Please give me your doubts and I will give answer. Yes, please ask me the doubts. Come on. Who dislike this video? Okay, so if you have any doubt, please tell me. Please tell me. Okay, repeat the concentration of substrate. See, the concentration of substrate is what? As you increase the concentration of substrate, the value of, you can say, the range of process or the speed of process will be increased, and that is a literal fact. If you have a 300 molecules of enzyme, this all 300 molecule of enzyme compose 3 sites, which is the active site. So it can be hold with the 3 molecule at a time. So 100 molecule of enzyme are capable to join and bind with the 300 molecules of substrate. But instead of that, what happened? All these 300 enzyme, they bind with the one of the substrate, and the process will finish in the second. Okay, so in the 1 second, what we get? We get only 300 molecule. Okay, the activity is about 300. 1 second, we get 300 molecule of product. If we get a 600 molecule of substrate, what happened? According to the 600 molecule of substrate, now every enzyme will bind with the 2 molecules of substrate. Clear? Every enzyme bind with 2 molecule of substrate at their active site. Maximum capacity is what? Maximum capacity is 3 molecule. So now at every second we get 600 molecule. When you increase it up to the 900 molecule of substrate, what happened? Now every enzyme, they bind with the 3 substrate molecule, because there is a maximum capacity of 3 molecules, right, with each enzyme. So activity now reach at the 900 points. Now when you increase it from more than 900 at the 1200 molecule at a time, what happened? That this value will be fall off. Okay, this particular value will be not fall off, but it will be statutory, because there is no space, because we have a 300 molecule of enzyme, and as we have a 300 molecule of enzyme, there is only and only 900 molecule can attach with this 300 molecule. So only 900 molecule will convert into the product every second. So the acceleration will be stopped at this particular point. So this was the idea, this was the core concept and the idea of our discussion today, right? So I hope you all the people likes this video. If you really like this video, and if you feel that this particular video was very helpful for you, please don't forget to like, share and subscribe it. Jai Hind.